The undesirable effect of enzymatic primary reaction causes losses to both nutritional and organoleptic quality, loss in texture, color, taste, phenol, vitamins. Therefore, there is need to stop this. But the question here is, how? In this video, we will be discussing about control and prevention of enzymatic primary reaction. For more video notification, subscribe to Easy Food Science and Tech. Heat treatment or application of heat. Our baseline here is optimum temperature of the enzyme, which is 40 to 50 degrees. At extreme heat application, the enzyme is totally destroyed. Processing methods that involve use of high temperature can cause the inhibition. A clear example is blanching of fruits and vegetables at 100 degrees Celsius. Note that time taken during the blanching or heat treatment is important. That is the blanching time, which depends on the size of fruit, type of fruit, and blanching method. During the blanching or heat treatment, some losses may occur in sensorial attributes or nutritional value. A classical example is during overblanching. To prevent the losses and concurrently inhibit the Brown reaction, is there any solution? The answer is yes. High hydrostatic pressure processing (HPP), a non-thermal approach that involves low temperature, which maintains the nutrient in the food. The principles involved is based on pressure-temperature relationship. Did you know that water can boil at 50 degrees or less? What matters is at what level of pressure. What does HPP do? It induces changes in the enzyme-substrate reaction or reaction mechanism. In the image, you can see how HPP causes changes in the active site of the enzyme, which the substrate cannot bind completely. If you can recall key and lock concept or model of enzyme, assuming the key is the substrate and the lock is the enzyme and the lock is damaged. For more video notification, subscribe to Easy Food Science and Tech. We have acid also, and our baseline is the optimum pH for the enzyme, which is around 5 to 7. Acid reduces the pH to low extreme point. Around 3 or below, acid totally inhibits the Brownian reaction. So, at high acidic condition, the Brownian reaction cannot occur. A clear example of this acid are organic acids. We have citric acid in lemon juice, we have malic acid in apple and grape juice, and we have ascorbic acid in orange juice. The next is antioxidants. Phenol compounds are stable, intact, or reactive in the apple, having a benzene ring attached with hydroxyl group, that is OH. When the phenol comes in contact with oxygen during peeling or cutting, the hydrogen atoms are lost, which results to the formation of unstable compounds for further polymerization up to the final products in the reaction, that is the brownish pigment melanin. Addition of antioxidants participate in inhibition or prevention of the Brownian reaction by donating the lost hydrogen atoms, thereby making the phenol stable and the reaction is arrested. An example of this, we have ascorbic acid, we have malic acid, and we have phosphoric acid. For more video notification, subscribe to Easy Food Science and Tech. For the enzymatic reaction to occur, it requires a helper molecule, that is cofactor, and is copper 2. It helps in giving the good conformation to the enzyme substrate complex. Removing copper 2, the reaction will not occur. And how? By using gelatin agent. An example is kojic acid, which bind or react with copper 2 to form a complex, thereby inhibiting the reaction. It's just like in brick block game that require a fragment to make a complete block to unquench. Then suddenly something come and stop it, meaning that the coins will not be earned. What stops the formation of the block is the gelatin agent, and the fragment required is the cofactor. So the reaction is arrested. The next is exclusion or removal of oxygen. When the tissues are not exposed to oxygen, that is, the gas we inhale, enzymatic reaction will not occur. It can only take place in the presence of oxygen gas if the tissues are exposed during harvesting, processing, and storage. Number one, we have use of packaged material that oxygen gas cannot pass through, that is, impermeable package. Next, we have water immersion, putting the sliced apple or yam in water, which prevent or reduce the reaction. Next, we have use of inert gases, that is, nitrogen gas and carbon dioxide. Next, we have salt and sugar solution, which will serve as coating to the tissues of apple or yam. For more video, subscribe to Easy Food Science